Good morning. It's April 30th, and this is your daily brief in science. Here's everything you need to know. A new study published in the journal eBiomedicine reveals a concerning link between phthalates, chemicals commonly used in plastic production, and heart disease mortality. The research from NYU Langone Health estimates that daily exposure to these chemicals was responsible for over 356,000 deaths globally from heart disease in 2018. Regions such as the Middle East, South Asia, East Asia, and the Pacific accounted for approximately 75% of these fatalities. Notably, di-2-ethylhexyl phthalate, or DEHP, emerged as a significant contributor, linked to more than 13% of heart disease deaths among individuals aged 55 to 64. The study highlights alarming disparities in health risks associated with phthalates, particularly in rapidly industrializing regions where plastic consumption is increasing and regulatory controls are often lacking. Researchers are calling for urgent global regulations to limit exposure, especially in areas experiencing heightened plastic production. The research utilized health and environmental data from various surveys across 200 countries, including urine samples that indicated chemical breakdown products from DEHP exposure. The economic burden of these deaths is estimated to range between $510 billion and $3.74 trillion. Future research aims to assess how reduced phthalate exposure could influence global mortality rates and to investigate other health concerns related to these chemicals. While the American Chemistry Council has not commented on the findings, they continue to advocate for the benefits of certain phthalates amidst rising health concerns. Dr. Leonardo Trasande, the study's senior author, emphasized the critical disparities in health impacts due to plastic pollution, challenging the notion that these issues primarily affect high-income countries. Don Pettit, NASA's oldest full-time astronaut, has just returned from a 7-month mission aboard the International Space Station, where he experienced a rejuvenating sensation due to weightlessness, feeling decades younger. Pettit, who has been an astronaut since 1996 and has flown to space 4 times, expressed interest in potentially flying again before he retires from space travel. During his recent mission, Pettit conducted extensive scientific research, focusing on areas such as metal 3D printing, water sanitization, plant growth in varying conditions, and fire behavior in microgravity. He also engaged in various physics experiments, including creating bubbles and forming a perfect ball of honey on a spoon with peanut butter. Throughout his time in space, Pettit took an impressive 670,000 photographs, driven by his desire to capture and share the beauty of the universe with those on Earth. He described his photography as a way to convey experiences that most people cannot witness firsthand, transforming memories into visual narratives. Pettit emphasized the importance of sharing his unique perspective of phenomena, such as meteors and volcanic eruptions, through photography, adding a deeper dimension to his stories. Upon returning to Earth in a Russian Soyuz capsule, Pettit faced the challenges of gravity for the first time in 220 days, which made him feel unwell upon landing in Kazakhstan. He noted that his return was difficult, as he didn't look or feel good, with his body's normal creaks and groans returning after the mission. Pettit credited his trainers and flight doctors for assisting in his recovery and readjustment to Earth's gravity. Despite his age, he expressed a youthful spirit, feeling like a little kid inside, and acknowledged that while he loves space exploration, there comes a time to return home. He also shared his passion for hands-on work, stating that astronauts often spend time repairing equipment, which he enjoys due to his love for mechanical, electrical, and fluid skills. MaaT034 is making strides in the field of cancer treatment with its innovative microbiome-driven therapies. Founded in 2014 and headquartered in Lyon, France, the company has been publicly traded on Euronext Paris since 2021. At the recent AACR Annual Meeting 2025 held in Chicago, MaaT034 unveiled new preclinical data for its product MaaT034, highlighting its potential to activate the immune system and combat tumors effectively. MaaT034 is a next-generation donor-independent synthetic microbiome therapy aimed at enhancing patient responses to immunotherapy. particularly when used alongside immune checkpoint inhibitors, or ICIs. In studies conducted on tumor-bearing mice, the combination of anti-PD-1 therapy with MaaT034 demonstrated a remarkable 83.7% reduction in tumor growth, in stark contrast to the 10% reduction observed with anti-PD-1 treatment alone. This innovative therapy not only reduces tumor growth, but also significantly boosts the production of beneficial metabolites, such as short-chain fatty acids, which are essential for improved gut health and immune response. In experiments with germ-free mice, 70% of the microbial species from MaaT034 successfully engrafted, indicating a stable presence of beneficial bacteria in the gut. MaaT034 is developed using a proprietary co-culturing platform designed for large-scale production and targeting specific oncology diseases. The therapy replicates the microbial functions of MaaT013, the company's lead asset currently undergoing a Phase 2 trial for metastatic melanoma. MaaT Pharma plans to present MaaT034 at several upcoming conferences, including the Swiss Biotech Day on May 5 to 6, 2025, and the European Hematology Association Congress from June 12 to 15, 2025. The AACR annual meeting where these findings were showcased took place in Chicago from April 25 to 30, 2025. Researchers at Washington University in St. Louis have made a significant breakthrough in spinal cord injury rehabilitation. They have developed a neural decoder designed to restore communication between the brain and spinal cord, which could enhance rehabilitation efforts for those affected by spinal cord injuries. The focus of the research is on creating a non-invasive brain-spine interface to assist in rehabilitation and enable voluntary movement in patients with spinal cord injuries. Led by Ismail Shanez and Carolyn Atkinson, the study demonstrated the decoder's capability to cue movement in the lower leg through non-invasive spinal cord stimulation, tested on 17 healthy participants. Participants wore EEG caps to measure brain signals while performing knee extensions and imagining the same movements. This setup allowed researchers to analyze brain activity and predict movement intentions based on neural signals. The decoder achieved an 83% accuracy rate in detecting actual movements and 77% accuracy for imagined movements. This innovative technology detects brainwave changes known as event-related desynchronization to trigger spinal stimulation at the right moment for intended movements. Notably, the system is non-invasive, using external EEG caps and transcutaneous spinal cord stimulation, unlike traditional invasive brain-computer interfaces that require surgical interventions. Sines noted that this approach could be especially advantageous for individuals with spinal cord injuries, where direct movement is not an option for data labeling. Future research aims to develop a generalized decoder that could utilize data from multiple participants, potentially streamlining clinical applications and enhancing rehabilitation efforts. Despite these advancements, challenges remain in achieving effectiveness with natural self-initiated movements, as current results are primarily focused on cued movements. The implications of this research suggest economic benefits through more effective non-invasive treatments, highlighting the necessity for updated regulatory frameworks as these technologies continue to evolve. The market for spinal cord injury rehabilitation technologies is significant, with around 18,000 new cases reported annually in the U.S., and rehabilitation costs can surpass $500,000 in the first year post-injury. The findings of this study were published on April 25, 2025, in the Journal of Neuroengineering and Rehabilitation, marking a notable advancement in the field. A recent study published in Nature Communications is reshaping our understanding of stem cell biology. Researchers have discovered that ancient genes, dating back over 700 million years, can convert mouse somatic cells into pluripotent stem cells. This collaborative research, involving HKUMed, Queen Mary University of London, and the Max Planck Institute, reveals that key stem cell genes previously believed to be exclusive to animals are also present in ancient single-celled organisms. The team successfully reconstructed an ancient protein known as WSOC from schwannoflagellates, demonstrating its ability to induce pluripotency in mouse cells, similar to contemporary SOX proteins. According to Professor Ralph Jauch, WSOC acts as a pre-adaptation, showcasing that molecular tools from unicellular life can effectively reprogram cells into pluripotent stem cells. These findings underscore the evolutionary importance of stem cells, suggesting they played a critical role in the emergence of multicellular life by enabling self-replication and differentiation into specialized cell types. This research challenges the long-held belief that only animal genes contribute to stem cell development, indicating that evolutionary tools could inspire innovative therapeutic designs. The ability of ancient SOX proteins to induce pluripotency opens new avenues for enhancing cell transformation methods for therapeutic applications. Future initiatives will focus on utilizing engineered proteins for conservation efforts and improving the quality of stem cells for therapeutic use. Insights from this study may also streamline disease modeling and propel stem cell research towards novel treatments and management strategies, significantly impacting regenerative medicine and addressing age-related health challenges. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.